Then we come to the immunoglobulins. अभी मैंने बार बार आपको कहा कि we will discuss it in the chapter of immunoglobulins. And what are the immunoglobulins? Immunoglobulins are the proteins which provide you immunity. Whenever antigen enter into a body, a specific immunoglobulin is stimulated. It combines with the antigen, and in this way the antibody antigen reaction takes place and your body is saved from the invading organism, bacteria or virus or whatever it is. Immunoglobulins are known as antibodies, are glycoprotein. ये याद रखना है यार. देखो molecular weight मैंने मना किया याद रखने से, but you should remember either it is a lipoprotein any molecule or a glycoprotein or a simple protein, right? They play part in the immune system by specifically recognizing and binding to specific antigen such as bacteria or virus and aiding their destruction. मैंने आपको बताया ना कि जैसे ही कोई बैक्टीरिया या वायरस इंटर होता है तो स्टिमुलेट करता है प्रोडक्शन ऑफ द एंटीबॉडीज, और वो एंटीबॉडीज फौरन उस एंटीजन से कंबाइन कर लेती हैं और उसे डिस्ट्रॉय कर देती हैं. And in this way, the immunoglobulins, they provide protection to your body. There are various immunoglobulin classes and subclasses which differ in their biological features, structure, टारगेट कहाँ करना है, किस ऑर्गन को टारगेट करना है, किस स्पेसिफिक एंटीजन को टारगेट करना है, एंड वेयर दे आर प्रेजेंट, दैट इज देयर डिस्ट्रीब्यूशन. स्ट्रक्चर, यार देखो हमने लाइट चेन्स की बात की थी ना, राइट? बेंजॉन्स प्रोटीन में, इफ यू रिमेंबर, antibody or immunoglobulin molecules are glycoproteins composed one or more subunits, each containing 4 polypeptide chain. 2 identical heavy chains, that is 2 are heavy chains, identical means similar, and 2 identical light chains. Heavy and light chains are held together by non-covalent interaction and also by the disulfide bonds. ये देखो, ये मेरा पॉइंटर देखो, this is the heavy chain, this is the light chain, ठीक है? And there is a variable region in both, and this is the constant region. And the variable region, it binds with the antigen. So each immunoglobulin is composed of 2 light chains and 2 heavy chains. And here you can see both of them, they are linked together by the disulfide bond. This is the constant region and this is the variable region. Purple one is the variable region. Constant region means the group or the composition, it remains constant here, whereas this change from immunoglobulin to immunoglobulin. FAB is the fragment of antigen binding. I have just shown you that it is the fragment of antigen binding, whereas FC is the crystallizable fragment, polypeptide protein sequence which differentiate the immunoglobulin on functional basis is present in this fragment. The amino terminal ends of the polypeptide chain show considerable variation in the amino acids. Obviously, they differ from each other, polypeptide chains, they differ from each other on the basis of the amino acid composition. वेरिएबल क्यों है, मैंने आपको बताया ना, वेरिएबल रीजन. आई गो अगेन टू द डायग्राम. वेरिएबल रीजन हमने इसको बोला है ना, व्हाई इट इज वेरिएबल. व्हाई इट इज वेरिएबल? बिकॉज द डिफरेंट, ड्यू टू द डिफरेंट अमाइनो एसिड कंपोजिशन इट इज वेरिएबल. The amino terminal ends of the polypeptide chain show considerable variation in the amino acid composition. and referred to as variable regions to distinguish them from the relatively constant regions. ये देखो कांस्टेंट रीजन है और ये वेरिएबल रीजन है. ये जो बॉक्स डब्बे 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 है ना, दिस इज द वेरिएबल रीजन एंड दिस इज द लाइट चेन एंड दिस इज द heavy chain. Each light chain consists of one variable arm, VL, L for light, and one constant domain, constant light. ठीक है ना? देखो अभी हम देखते हैं इसको. देखो ये वेरिएबल रीजन है, ठीक है. ये लाइट चेन है. चलो लाइट पे हम बात करते हैं. VL and VC. This is the constant region and this is the variable region. Heavy chain के लिए दिस इज द वेरिएबल रीजन और यहां से यहां तक का ये कांस्टेंट रीजन. ईच हैवी चेन हैज अबाउट ट्वाइस द नंबर ऑफ अमाइनो एसिड्स. यार वो लंबी भी तो कितनी है ना, मॉलिक्यूलर वेट भी ज्यादा है, नंबर ऑफ अमाइनो एसिड्स भी ज्यादा है, ठीक है. A chain consists of a variable domain VH, सही है, and three constant domains CH1, CH2, CH3. ये वेरिएबल डोमेन है इसका और ये इसका कांस्टेंट डोमेन है. And here you can see it is double in size. Double in size है, obviously the amino acid number will also be large. The V region, variable region. V for variable. The V region of H and L chain comprise of antigen anti-binding sites of immunoglobulin. Each immunoglobulin, दोबारा से जाएंगे डायग्राम पे. ये this is the antigen antibody region. Here the antigen binds to the immunoglobulin. Each immunoglobulin monomer contains two antigen binding sites and is said to be a bivalent. The hinge region in the area of the H chain between the first and the second C region domain is held together by the disulfide bond. Again I am going to the diagram. This hinge region. Here it is linked together by the disulfide bond. Here also there is a disulfide bond. The flexible hinge found in IgG, IgA, IgD, but not in IgM or IgE. Region allows the distance between two antigen binding sites to vary. There are 5 types of immunoglobulins: IgG, IgM, A, D, E, and they are distinguished by the type of the heavy chains which are present in them. IgG molecule have gamma chains, have mu chains, have alpha chains, silent chain, delta chains. Different types of chains are present. Difference in heavy chain polypeptide allows the immunoglobulin to function in different types of immune response. Heavy chain different है and therefore the immune response will be different. The polypeptide protein sequence responsible for these differences are found in the constant fragment. It is not found in the variable fragment, it is found in the constant fragment. That is the area which is responsible for different type of immune response is present in the constant region, and therefore the one immunoglobulin differs from the other immunoglobulin due to the difference in the constant region. Antibody classes. differ in as a result of different number of Y units, monomers, which are arranged together. For example, IgM, it has a Y-shaped units arranged in a pentameric form, containing a total 10 light chains, 10 heavy chains, and 10 antigen antibinding sites. IgM, pentameric form. Okay? IgA, dimeric form. And these are the monomers. IgG, IgE, IgD, these are the monomers. Whereas IgM is present in the pentameric form, and they are arranged together by the disulfide bond. Whereas IgA is a dimer. IgM pentamer, that is the monomeric units, they are joined in a pentamer. Okay? It is present on the mature B cells. First antibody which is produced in response to an antigen. First antibody produced by the fetus, efficient in binding antigen in many repeating viruses and other, and they carries out a complement activation. I have shown you what is complement activation, how it takes place, what is the importance of this. You can revise your previous lecture. Then IgD, monomer, membrane-bound immunoglobulin on the surface of the mature B cell. No biological effector function is known. IgA, predominant antibody present in the secretions of saliva, tears, breast milk, and mucosa. And it also provides a first line defense against infections. IgG crosses the placenta. It can be found in the newborn, and most abundant of its types is IgG1, IgG2, IgG3, IgG4. IgE provide defense against parasitic infection. It is a monomer, you have just seen. Only two are the one is IgF, which is a pentamer, and other is the IgA, which I have shown you, it is a dimer. Defense against parasitic infections associated with hypersensitivity reaction and found mainly in the tissues. Opsonization is the coating of the particle with protein, and this coating, it facilitates the phagocytosis of the particle by the tissue macrophage and facilitate the binding by receptors on the peripheral blood cells.